I have the privilege to introduce to you the plenary speaker of today, Roman Stocker. And Roman Stocker studied up with a PhD in civil and environmental engineering at Padua in Italy, and then he went on to Western Australia for a short while, to Perth, University of Western Australia. And then he moved to the MIT, to the Department of Civil and Environmental Engineering, and he is since then trying to make the invisible visible, and using his education as an environmental engineer and looking into biological systems, and he comes up with really amazing features which you barely and you usually don't see, characterizing essentially microbes in their environment, how they experience being a microorganism, how they experience microenvironment, nutrients, something what we usually filter some liters of water to determine nutrient concentrations. He knows better how this is distributed in a drop of water. Please welcome Roman Stocker.
Thank you for the flattering introduction, Gerhard. Sorry for the slight delay here. To the organizers, thank you for this opportunity. I'm truly honored, and thank you for bringing us all together in this wonderful city of Granada. I would like to tell you about ocean microbes, but I would like to do so from a somewhat unconventional angle, one that emphasizes the microscale, that emphasizes direct visualization of microbial processes, and that integrates physical concepts into the understanding of the ecology of marine microbes. I'll be talking about microbes that are marine, but I think you'll be easily able to port over the same concepts to microbes from other aquatic environments. The talk will be in the shape of an hourglass. I'll start broad by introducing the key microscale players and processes through brief examples. This introduction will allow me to distill the concept of ocean biophysics and to describe why microfluidics and video microscopy are ideal tools to look at these small scales. And then I'll broaden out again and look at three applications of these approaches. I call them close encounters, in the sense that they will look at direct interactions at the microscale between different biotic and abiotic constituents of this microscale world. And then time will run out. One of the difficulties of studying microscale processes in the ocean and communicating their importance is their sheer invisible scale and the fact that we lack an intuition for what their world looks like. So in order to give you a flavor of what the world of marine microbes look like, I'd like to invite you to come on a short dive, except that we'll make it no ordinary dive. Imagine that on this dive you had a special mask that allows you to magnify things at will. What would you see? I think you would see a myriad of different things, and it would be an intriguing adventure. I'll only have time to introduce you to a few of them with the aim of introducing the main players, to give you a sense of their size, abundance, and importance, and then to give you a sense of what their environment looks like at small scales. This is not going smoothly. There we go. Thank you. You would almost certainly see some of the most abundant photosynthetic organisms on the planet. These are the micron-sized Prochlorococcus. They're less than a micrometer in diameter. There is a staggering 10 to the 27 cells in the ocean. Cumulatively, they have the same net primary productivity as global croplands. Even more abundant are heterotrophic bacteria. There is 1 million of them per milliliter, and this is the total number that you get in the ocean. They play an essential role because they recycle dissolved organic matter. They're essentially the only organism can take up solutes and return them to the classic food web by producing biomass. And they serve a number of other purposes, including, for example, carrying disease to corals. Some of these heterotrophic bacteria swim, and they do so by exceedingly thin and long flagella that allow them to achieve propulsion. Killing both of these types of organisms are abundant viruses. These are 10 times more abundant than bacteria, They are now hotly studied in oceanography. A lot remains to be discovered about their species specificity and ecological strategies. For the purposes of this talk, what matters most is their minute size. They're typically smaller than the smallest wavelength of light, so they're the ultimate challenge for dynamic visualization. And then you would see a broad range of things that are not alive, but either derive from things that were alive or that have intense populations of microbes that colonize them or consume them. In particular, you would see particles of different sizes, shapes, and compositions. You would see them with your special mask that they slowly settle, and you would understand why they're called marine snow. The sinking induces significant transport of carbon from the upper ocean to depth, a process called the biological pump, which is arguably one of the most important carbon fluxes in the ocean. The full importance of these players and processes has not been realized all too long ago. It was thought until the 1970s that heterotrophic bacteria were few and not very effective at taking up carbon. Our current thinking stems from a quiet revolution led by the likes of Larry Pomeroy, Farouk Azam, and other giants in the field. And what emerged was a concept of the microbial loop, which is indicated here by the yellow loop of arrows. Essentially, vast amounts of carbon are transformed through a range of processes into a dissolved pool, DOC. And it's only thanks to bacteria that they're routed back to biomass and therefore to the classic food web. But what I have just shown you is a static view of the microbial loop that emphasizes players, interactions to some extent, but makes no mention of the dynamic component of this environment, in particular temporal timescales, as well as spatial heterogeneity. And if you think about it, ecology is all about time and space at the scale of the organisms. So I'm afraid we'll have to put on our mask again and go for a second dive and try to see whether we can get an understanding of what the environment really looks like at these small scales. And in so doing, we'll also discover that there is a broad range of physical processes that drive many of these microbial interactions. Perhaps the dynamic feature of the ocean environment that most stands out is cell motility. Some bacteria do not swim, but many others do, and they will be zipping past your special mask at a striking speed of tens to hundreds of body lengths per second. Motility is also pervasive among phytoplankton. 90% of species producing red tides, for example, are motile. Motility is used, for example, by predators. This little Bacteriovorax cell giving a chase to Vibrio coralliticus. Once it finds it, and you will never find out from that video because they disappear, it bores into it, it replicates inside its prey, and then it kills it. Even cells that are non-motile move around. This Prochlorococcus cell and its viruses jiggle around by a physical process called Brownian motion, which results from the incessant collision with water molecules. This is much slower than motility, but by no means unimportant. The reason for that encounters are always based on relative motion. And if it weren't for Brownian motion, these two things would rarely encounter each other, as we will also see later on. Death can also be a very dynamic process, as the lysis of this diatom chain demonstrates. This is produced in the laboratory by intense zapping of light, but these lysis process of diatoms also occur all the time in the ocean due to a range of processes. In this case, you see this mini explosion happening and then the diffusive release of the contents of the diatom into the surrounding water. There are plenty of heterotrophic bacteria waiting for these little hotspots out there. They can use chemotaxis to take advantage of that. Chemotaxis is the ability to sense chemical concentrations and direct motility accordingly. In this video, we see another diatom, and the black dots are individual bacteria. We see a low level of accumulation around the body of the diatom, which, when it lyses, induces, right now, a massive frenzy of bacteria. They all move in by chemotaxis in order to consume the chemicals that have just been released by the lysis. You see time up here, and you can see it's sped up considerably. Now is that bacterial frenzy again. They move in and consume a large fraction of it. We can quantify the spatial distribution of microbes in these movies very accurately, and therefore plot the concentration of bacteria relative to background. And the logarithmic color scale here gives you a sense of the 3 order of magnitude increase in bacterial concentrations in that particular ecological hotspot. The prevalence of ecological hotspots produced by chemotaxis is really widespread in a number of different scenarios. I already mentioned the association with phytoplankton up here. We see them around this dead copepod in the form of these light gray trajectories, and a few years ago we have demonstrated that they also cluster inside the plumes of sinking particles. And I'm still grateful to ASLO for recognizing this work at a time with the Lindemann Award. It meant a lot at a time in which we were just getting started with that research. And I take this opportunity to salute the young scientist who has won the Lindemann Award this year. Well done. Physical element that is prevalent in the environment that microbes live in are interfaces and surfaces. Here, for example, you see the case of the interface between an oil droplet and bacteria and how strongly they have accumulated right around at that interface. This picture single-handedly changed my view of how bacteria interact with pollution in the environment. We're working on this aspect quite dynamically. I won't have time to report too much on this. Intriguing and important surfaces that microbes interact with are the surfaces of larger animals, such as corals, for example. If a bacterium had eyes, this is possibly the view of a coral that it would have. And I will tell you a little bit later what the physical environment looks like due to the presence of these cilia on the surface of the coral. A few years ago, we've put together this image out of these concepts. I refer to it as a microbial cube, and it means to show something like a milliliter of seawater, but not just the players, also the dynamic interactions as well as the spatial component of this landscape, with marine snow particles that sink, bacteria that give chase to these particles with chemotaxis, excretions by larger organisms, lysis and leakage events. And since you'll see me show a few of these graphics, I'd like to spend a couple of words on them. We have found them to be a very powerful vehicle to communicate this otherwise invisible world also to broad audiences. And we've been fortunate to work together with a graphic artist in the UK by the name of Glyn Gorik. So I wanted to acknowledge and thank officially Glyn for all the hard work that is put into the collaborations with us. I've kept one physical element that is prevalent in the microbial world for last, and that is fluid flow, and in particular turbulence. It's hardly a new concept that turbulence influences plankton. It was pushed very hard already by Ramon Margalef himself. But there is now a renewed interest, particularly at the microscale, with an influx of ideas and tools from physics and engineering. For example, a counterintuitive prediction is that turbulence may actually unmix populations of motile phytoplankton, producing strong patchiness and therefore changing the encounter rates between organisms. But this is a very young field and lots remains to be tested and asked in this area. Laminar flows can also have surprising impacts on motile organisms. This bacterium undergoes what is known as Jeffrey orbit. The velocity gradient in that flow exerts torques on the organism that continuously reorient it and therefore change its swimming trajectory. Far from just being a local effect on the trajectory, these effects can significantly trap bacteria, for example in regions of high shear, therefore hindering important survival strategies such as chemotaxis and favoring others such as surface attachment. Phytoplankton can also be trapped by flow in their daily commute upwards when they encounter regions of shear that are too strong for them to maintain a stable vertical orientation. And that results in them tumbling heads over heels and accumulating at certain depths. A few years ago, we've proposed that this might be one of the mechanisms that creates thin phytoplankton layers, which are ecological hotspots frequently observed in the coastal ocean by oceanographers, and that in some cases can be precursors of red tides. I believe we're just beginning to understand how rich the interaction between flow and microbes is. So for those of you who are out there looking for questions, young scientists, I invite you to look at the interaction between flow and microbes as a very rich topic of research. You're changing the screen on me all the time. We'll just leave it as it is, even if it's black. Thank you. This long introduction allows me to now mention what I mean by ocean biophysics. I look at this at the study of microbial processes in marine and aquatic environments, at the spatial scale of microbes, at the temporal scale of microbes, in particular taking explicit account of their temporal dynamics and capturing the dependence on the chemical and, most importantly, the physical microenvironment. The tools that we have found most suitable to study these processes are a combination of imaging and control of microbial environments. Imaging is typically done at this scale by video microscopy. You see an objective of a microscope here, as well as image analysis. And in the last 10 or 15 years, microfluidics has really afforded unique possibilities to control microbial environments. You can fabricate these small chips in the laboratory with great accuracy and not great cost. You can use them to set up chemical gradients as well as fluid flows, all the while imaging directly what the response of microbes is to these cues. So what I'd like to do in the last part of the talk is to give you 3 examples of these approaches. Each one deals with a microbial encounter and its implications. Because they're small scale, we can call them close encounters. Those of you who are science fiction fans know that this also obeys the definition of human observations of actual animate beings, so they're legitimately called close encounters of the third kind. So in the last part, I'll tell you about 3 close encounters of the third kind we've had recently in the lab. The first one deals with the encounter between viruses and hosts. This is a project led by PhD student Kwang-Min Sun in collaboration with the groups of Matt Sullivan, Pelly Chisholm, and Jeff Waston. These are 2 traditional views of viruses. On the left, you see them stuck on a filter and stained, probably for counting. On the right, you see an electron micrograph, which has much higher morphological resolution. But both of these are static pictures that fail to capture the dynamics of a virus as it encounters its host, it moves in, it sticks or does not stick to the host, and then eventually infect it. So Kwang-Min has recently succeeded in obtaining a dynamic view of some viral processes. He calls this viral video. He combines microfluidics to have a careful observation environment with video microscopy. Our model system is Prochlorococcus and one of its viruses. Prochlorococcus is naturally autofluorescent in red because of its chlorophyll, and the myovirus we stain with green fluorescence. Kwang-Min then uses an image splitter such that half of the camera chip contains the image of the viruses and the other half of the camera chip contains the image of the host. He then puts them together and you can see what you get. On the right you have a view of hosts and viruses, Prochlorococcus is in red, and on the left you have the same video after image analysis and tracking have been applied. So you can see from here now that we have full information on their dynamics. We know their positions, we know their trajectory, and we know their relative locations. Now, if you look into the oceanographic literature, you find that the consensus seems to be that most contacts between viruses of bacteria and host cells result in adsorption. Quantitatively, that would mean that the adsorption efficiency is in the order of 100%, meaning every encounter should result in an adsorption. That seemed questionable to us, at least from the point of view of the fact that the receptors are really discretely located on a cell surface. So we decided to take a quantitative look at this. But let's go in order. Before we have an adsorption, we need to have an encounter. And we already said that the 2 players here are both non-motile, so the only way to encounter each other is by Brownian motion. From the trajectories, we can very carefully quantify the diffusivity due to Brownian motion of both the host, H, and the virus, V. And then we can resort to encounter rate theory to predict the encounter rate, which is proportional to the sum of the diffusivities, how much they jiggle around, the sum of their radii, they need to come in contact, and the product of their concentrations, as is typical in encounter theory. This gives us the prediction here by the maroon bar on the left, that the encounter rate is about 1 times 10 to the minus 9 milliliters per minute. We can now go into the viral videos and score each and every encounter, divide by the measurement volume, divide by time, and we obtain an experimental estimate of that. And we see that the 2 are in good agreement. We then did a control experiment in which we took another strain of Prochlorococcus that is known not to be a host for that virus, and we found again the same number, which is reassuring because encounter rates are purely a physical process. But Kwang-Min can go a lot further with this, and he can zoom in on individual host virus pairs. On the top left you see the original movie, in the middle you see the tracked trajectories, and on the right you see the distance between host and virus in micrometers, where 0.7 is the threshold for detecting an encounter. In this particular case, we get an adsorption. You see they start far apart, they come closer and closer, and then they remain close enough for a sufficiently long time. But by no means all encounters result in adsorption. Many result in an escape, such as this one. You can see when the movie loops that they start again far apart. This is the first encounter, the second, and the third, and yet the virus moves away, and this does not result in adsorption. When we do this over thousands of trajectories, we find that only about 5% of the viruses are adsorbed. All the other ones are escaped. When we do it for the non-host case, we get 100% escape, as you would expect. To make sure that these numbers are approximately right, we applied a second independent approach, counting free viruses, meaning viruses that have not yet adsorbed to hosts. If you go to theory, it predicts that the logarithm of the number of free viruses, V, should decrease linearly with time. And this is precisely what we observe. And this curve then allows us to back out a second estimate for the adsorption rate, K. Importantly, this is a non-invasive approach. We never disturb our sample because we count by imaging, and we check that no viruses are attached to the microfluidic boundaries. If you put the two methods together, you see that there is very good agreement between the two adsorption rates, and most importantly, the adsorption rate is much smaller than the encounter rate, which already tells you that their ratio, the adsorption efficiency, cannot possibly be in the neighborhood of 100%. In fact, when you do this very carefully with all the appropriate controls, including controls for the fact that we stain the viruses, you find an adsorption efficiency of about 4%. When we repeated this at the macro scale with the classic adsorption kinetics assay, we found a value of about 40%, a full order of magnitude larger. What that tells us, or our interpretation of this is that the invasive step in this approach, often centrifugation or filtration, provide plenty of opportunities for viruses to stick to extraneous surfaces, and then they are incorrectly counted as adsorbed to the host, and this skews this value upwards. If this bias is true across all systems, it would imply the need to revise not just the adsorption efficiency that we estimated, but also the infection rates, because adsorption is a direct precursor of infection. What I just shown you is a case of a passive encounter driven by Brownian motion. Let me now switch to a very active encounter in which the bad guy, here a coral pathogen, encounters its host by motility. This is a project driven by two postdocs, Melissa in my lab and Or in the lab of Asaf Vardi at the Weizmann Institute. Here we're focusing on the last millimeter of seawater directly adjacent to the surface of a coral. It's a chemically very rich environment because of the coral mucus that the coral continuously secretes. So we hypothesized that coral pathogen might be able to sense these gradients and swim towards them by chemotaxis. To test this, we built a microfluidic device where we created a 400 micrometer wide layer of mucus, similar to what exists in the environment over coral surfaces, and we exposed a population of pathogens to that layer of mucus. The graph shows you what we get when we image the population and plot its distribution across the channel width as a function of time. You can see bacteria out here initially, and then you see this massive wave of bacteria that move into the mucus layer. The strength of this response caught us by surprise. We quantify these chemotaxis experiments always in terms of this chemotactic index, and we see here that we get values in excess of 10 typically. To put this in perspective, the maximum value we ever got for E. coli, which is a model system for chemotaxis, is about 2. So very strong, very fast chemotaxis towards these chemicals. We also know that the chemical responsible for this seems to be DMSP. When we repeated the experiment with 15 micromolar DMSP, which is the concentration that is in coral mucus, we find essentially the same attraction, the green line here. We then concluded, they must be swimming towards the DMSP and also probably taking it up, because it's otherwise a rich source of carbon and sulfur, but lo and behold, they don't seem to care. Nuclear magnetic resonance imaging showed that Vibrio coralliticus does not metabolize DMSP. And that led us to conclude that DMSP is purely an infochemical. It's purely a signal that allows them to get to their host. Things get worse for the coral once they are stressed by increasing water temperatures. We went out to Heron Island on the Great Barrier Reef, and we temperature stressed some coral fragments. We took their mucus, we shipped it back to MIT, and we repeated these experiments. And what we found is that the pathogen responded twice as strongly to the mucus from the stressed corals, and that this was hand in hand with a 5-fold increase in DMSP in the stressed mucus. But one question kept nagging us: why the hurry? We were surprised by this rapid response, and at least compared to my naive interpretation, it sounded strange. I thought of the coral surface as something that is a vast expanse of coral mucus where the coral pathogen could simply swim in. So that led us to zoom in much more closely to the surface of the coral, and this is what we saw. The impurities here allow you to broadly trace out what the flow is doing. We can process this image, and you see it's not a smooth environment, but it's a strong vertical flow. And this is not a fluke either, but you see these patterns everywhere on the surface of the coral. Now, in addition to being important, as I will show in a second, these flows are also quite beautiful, I think, as is fluid mechanics quite often. We can take a next step and trace or track these impurities in order to get information on the velocity field. And from this we learn two things. The first thing is that these flow speeds are much, much faster than the swimming speeds of the coral pathogen, which is typically less than 100 micrometers per second. So they're really in a storm here. The second point is that the maximum velocity are right adjacent to the surface of the coral. So this is incompatible with any explanation that suggests that these flows are caused by an external flow that then interacts with the topography of the coral. Instead, the solution to this is that it is the coral itself that causes this storm. This is a video taken at 1,000 frames per second that shows the beating of the cilia on the surface of the coral. This is no different than what is happening to each one of you out there right now in your airways, where cilia move mucus and impurities out. Except that in this case, it's done in a vertical pattern. If you zoom in with an SEM, you see how dense this canopy of cilia is on the surface of the coral. And from here, you also see the boundaries between individual cells and these intriguing anchoring points. These are truly amazing structures. These flows are very robust and they're also quite steady. Here we're imaging the 500 micrometers between two polyps. The orange and cyan streamlines here are the flow imaged at interval of 90 minutes. You see small shifts due likely to these pulsations of the coral, but by and large the vortex is still located here an hour and a half later. By pulling in water and pushing out water, these flows have major effects on mass transport and oxygen transport, as we found from a mathematical model, where these are some of the predicted effects. Let me take this back very briefly to our coral pathogens. I told you that they're exquisitely able to swim in towards the coral mucus, but also that they encounter a very challenging physical environment once they get there. So Doug Brumley, a postdoc in the lab, is now working at trying to track the swimmers in this coral storm. And to do that, he uses what he calls a coral disco, where he confines a little coral fragment in a microfluidic device. He adds blue fluorescent beads as well as red fluorescent bacteria that are courtesy of the Vardi lab. And then he uses dual challenge imaging, where he fast switches between two LEDs, one for the beads, one for the bacteria. So very preliminary results from the action inside the coral disco are the streamlines here and the swimming trajectory of the bacteria. And you can immediately see that the bacteria can best swim in regions where the flow is rather slow, down here and in the eye of the vortex. But next to the coral surface, where the flow is faster, they are primarily advected by the flow. So a pure speculation at this point is that these flows may serve an additional purpose, which is to act as a sort of simple physical immune system and keep at bay invaders. This takes me to the last part and the last close encounter, which deals with the degradation of marine snow. It's a project led by Gabriel Juarez in collaboration with Martin Pols and Jan-Hendrik Hegemann. If you look into the literature, it's rather surprising, or it was to me, how little is known quantitatively about the microbial degradation of particles, in view of the fact that they really represent an important flux in the biological pump. So much so that most models assume first-order kinetics in the absence of better information. So we asked the question, can we directly visualize degradation and from this perhaps come up with some more robust models? So Jan-Hendrik fabricated small alginate particles, which Gabe then put into a microfluidic device for observation. He exposed them to a Vibrio population for a short amount of time to get some initial colonization, and then he washed away the remaining microbes. This gives the initial condition that is the start of this video, which will be a time-lapse confocal microscopy video. The real duration is about 24 hours. So after a full morning of talks, I had to do something to keep you awake for the last 5 minutes of my talk. What I think you saw is the growth of those initial colonizers into little colonies. Not much happened to the particle up to that point, and then they started munching away at the particle, taking off little bites, and then excavating it until the whole thing crumpled. And the final thing that is left is this thing, which we now know contains no longer any alginate, is simply a ball of bacteria. Here's another view of that for 3 different particle sizes, this time in 2D and without any music, and you'll see exactly the same processes happening. From videos like this, we can now very accurately quantify the size of the particles as a function of time, and we do that by calculating the equivalent diameter, normalizing by the initial diameter. And you can immediately see from this that the degradation kinetics are certainly not linear. In fact, there is a long lag phase here, followed by a very swift death of the particle. It gets consumed essentially within an hour and a half, and then this clump of bacteria is left. There is, by the way, an interesting puzzle here, which we haven't solved yet. The final diameter of that bacterial clump is always 50% of the initial diameter of the alginate particle, and there it stops. We don't know why. These observations allow us now to propose a physically based model for degradation kinetics. We're interested in predicting how the mass of the particle changes with time. And we write that rate of change as the degradation rate per cell, how much each one consumes, times the number of cells on the particle at that time. And that we in turn write as the initial number of cells times exponential growth. And we have evidence that growth on the particles is indeed exponential. We then need to take into account some inefficiencies. Not all of the alginate that gets solubilized becomes biomass. Some diffuses away in the environment, some is respired. So that gives us an efficiency epsilon. And finally, this results in a prediction that we can test against experiments. When we do that, we get very good agreement with our observations. There is still one fitting parameter here, which is that efficiency epsilon. We find a reasonable value for it, but we're also working at trying to constrain it independently. So what this tells us then is that it is the growth of the bacteria on the particle that is responsible for the nonlinear degradation kinetics. And we're now in the process of implementing this formulation in a marine carbon cycle model with Curtis Deutsch from UW to see when, if and when it makes any difference compared to current formulations. In closing on this project, I find it fascinating that some of these fundamental mechanisms might carry over to completely different situations. This is a droplet of crude oil, about 100 micrometer in size, suspended in a suspension of oil-degrading bacteria. You see some initial colonizer on the surface here again, highlighted, and if I run the video, you see the growth of these initial colonizers up to the point when they completely cover the surface, and only then is there some action on this little droplet that completely loses its shape and again crumples. So they're close analogies with what we saw with the marine snow, despite the fact that that was a solid particle and this is a liquid-liquid interface. In closing, I'd like to reference a book written by E.O. Wilson in 1998 called Consilience, in which he advocates for greater unity between the different branches of our knowledge, including humanities, science, and the arts. At a much smaller scale, I'd like to argue for increased marine consilience and for the need to continuously look at the interface between different disciplines, bring them together, and therefore discover more. I found that the angle of mechanisms, dynamics, and single cells is particularly conducive for entraining people also from different disciplines. In particular, I'm often exposed to physicists from soft condensed matter physicists and applied mathematicians, and they find a nice point of entry into these problems via this mechanism. I am aware that I have squarely focused on the microscale, and that a challenge as well as an opportunity ahead is to foster a better handshake with the macroscale. Ultimately, the situation we'd like to be in is one in which we understand microbial scale mechanism and ecosystem scale implications at the same time. So I'll close here. I'd like to thank funding agencies, NSF, NIH, the Human Frontier in Science Program, JGI, and the Gordon and Betty Moore Foundation, but above all, the current and past members of my group, to whom much of the credit of this work actually goes, for their skill, their drive, and their passion in pursuing this research together. Thank you very much.